to do the Quizlet or for the final?
Nothing for the final. Maybe, I guess I could maybe make one. There's just so much material. So there's, I think for all of the old Quizlet or all the old, yeah, Kahoots and quizzes are up there. Yeah, so I would say go over those. I don't think it's going to do a whole lot of good to make a new one based on, yeah, because I'll probably just reuse some of those old questions. If you do want to see any of your old tests, I'll bring those in Monday. I don't know if I mentioned that. So for you guys who are on Zoom, if you want to see your exams, and you want to come in Monday, then we'll see how many people fit in here. And if it's too many, we'll just go over to the bio labs. So I'll bring all of the old exams so you can look over them. And then we'll do like the exam 5 review, if that makes sense. Or if you can't come Monday and you want to see your exam some other time, that's totally fine. Just email me or come by during office hours and I'll have them. Okay, what am I doing? So these last couple chapters are pretty big picture. We were talking about biotechnology, so some more applied stuff, which is kind of cool. And then genomics, a lot of which is applied and also is really closely related to biotechnology. And I'm going to sort of focus on that aspect of it, like how this is applied to biotechnology and mostly medical stuff in this chapter. So yeah, let's just get started on genomics. So what is a genome? I think I've mentioned the word genome before, and all it is all of your DNA, all of an individual's DNA, all of the genetic material within a person. That's a lot. And we're going to talk about the Human Genome Project, which actually determined all of the DNA, all of the sequences essentially in human beings. There's a couple different maps you can make to sort of map a genome. Genetic maps and physical maps, I'm not going to get into too much detail. Basically, genetic maps I'm not going to talk about anymore. We've sort of referenced these when I talked about recombination frequency. So the more likely genes are to kind of recombine with each other, you can kind of figure out where they are in relation to each other on a chromosome. If they recombine a lot and aren't often found together, they're probably pretty far apart. And then vice versa, if they're often found together and linked, they're probably pretty close to each other on that chromosome. That's the idea behind a genetic map. You have to look at offspring frequencies. So you have to actually breed something and then look at, what the genetic or the phenotypic makeup is of that offspring. Physical maps are sort of how things are done now in genomics. It's a precise location, so you can kind of map out, based on map units, the precise location of genes in a genome. So you know what chromosome they're on, you know exactly where they are on that chromosome. It's not relative to other genes. It's exactly like a physical place on that, in that genome. The ultimate physical map is basically the complete DNA sequence of a genome. So what do I mean by a DNA sequence? They have the ability now to look at all of the bases. So the A's, the G's, the T's, the C's, every single one of those in an individual, whether it's a fish, a dog, a person. They can look at this entire base pair sequence. That's what we consider the ultimate physical map. You know, each and every nucleotide that's there. All of these sequencing approaches. So when we say sequencing, it's just what sequence are those letters, those nucleotides in? What's the code, essentially? They all take DNA replication and then put it in a test tube. So before they were using bacteria to replicate the DNA, they had to wait for the bacteria to reproduce, and it took a little bit longer. Now they can do it all in a test tube or in vitro, a lot faster and easier in a lot of ways. Technology in this area has moved along probably quicker than technology in any other area of at least like basic biology. So the advances in genomics over the past probably 20 years have been astounding. I'm going to talk a little bit about those now. It's obviously not easy to look at all of the base pairs in a genome. So a lot of new technologies have come about to make it easier. The general category of what's used now, and you might hear this term at some point in the future, but it's called next generation sequencing or next gen sequencing. And I'm not going to talk about any of the others just because this is primarily what's used nowadays. The speed and the speed has increased massively. So how quickly you can actually sequence a genome has changed a lot. And then the cost. So the cost has come down massively as well. In 2014, well, compared to, 2004 to 2014, there was basically a 10,000-fold reduction in cost. So you can sequence a genome in 2014 for about $1,000. Before that, it was 10,000 times that. So the technology has advanced quickly, which makes it faster and cheaper to be able to sequence genomes. There's a few different kinds of next-gen sequencing technologies. I'm not going to get into detail about too many of them. I'll give you an example of one on the next slide, one that's pretty commonly used, but they all have a couple of features in common that you want to be familiar with. So they essentially can look at millions of sequencing reactions at once. So when they're trying to match up, what letters are in the genetic code, they can do all that at one time. They don't have to do it sequentially. And then you can directly read the sequence of bases rather than, if you remember the gel electrophoresis technique I mentioned, that separates out DNA based on fragments. So you're not actually looking at the A's, the G's, the T's, and the C's. You're looking at the size of the fragments. So you're looking at something different, and you can get a lot more information if you know the actual bases. So now next-gen sequencing, you can look at the bases within the genome rather than just the fragment size and then try to kind of match that up, which we didn't talk a whole lot about that process, but this is a lot more effective, efficient, and direct at figuring out what the sequence is of a genome. So the one example I want to talk about, hopefully this will help you at least get a better idea of what next-gen sequencing does, is called Illumina DNA sequencing. This is where they take a single strand of DNA, so they basically unzip some DNA. They can manufacture the nucleotides, the A's, the G's, the T's, and the C's. They put those in a test tube with this unzipped DNA and see essentially what letters match up, right? So what nucleotides are used. Then you can tell what the other letter is going to be based on the base pairing rules. So you can determine the sequence of bases just by cutting DNA in half, adding in nucleotides, and then they read that with a laser. So they color code these nucleotides. So A is a certain color, C is a certain color, etc. So then they essentially read using a laser, they'll read those bases that they added and see what the pattern is. Then they can see what the complementary base would be, and they know the sequence of that DNA pretty simply and pretty quickly. So they can do a lot of DNA molecules at once. They don't have to just do one or two. So it's a really effective way to do it. They just look at what bases are added, and then that gives you the genome sequence. So that is Illumina DNA sequencing. And the Human Genome Project. So this is one of the applications of genomics. In 1990, they started a project called scientists around the world started a project called the Human Genome Project, where they wanted to sequence the entire human genome. So look at all of the base pairs, all of the genes in the human genome, which wasn't known prior to 1990. And they wanted to know the function of all the genes too. That's kind of still ongoing. They started in 1990, finished in 2003. So it took them 13 years to complete this. And it cost $2.7 billion. So it's crazy expensive, and it took a really long time to get one sequence done, basically an individual sequence. So now, like I said, the technology has come a long way. It can be done in a few hours, so you can sequence an entire human genome in a few hours for $1,000. So it's amazing how far we've come as far as genomics in the past, few decades. So the Human Genome Project was still really beneficial. More and more has been done with this, obviously, with the increase in technology. So I'll talk a little bit more about that. But some of the cool outcomes of the Human Genome Project, I wanted to focus on just a couple that were particularly interesting to me. So before this project was completed, scientists thought there were about 100,000 genes in the human genome. Really, we only have 20,000. So we have far fewer genes than we originally thought. I guess we thought we were more gene rich than we actually are. It's still 3 billion base pairs. So that's why it took 13 years with the older technology to figure out what all of those base pairs are. But 20,000 genes. And it turns out that's about the same number of genes that mice and pufferfish have as well. So among probably other species that they've sequenced since then. So we have the same number of genes as a mouse and as a pufferfish. And some simpler organisms have far more genes than we have. So if we look at this graph down here, I'm just going to focus on the y-axis, which is just the number of genes. You can see humans are over here on the right. So for you guys on Zoom, I'm focusing on the y-axis. So number of genes, humans are all the way over here on the right. Oh, right here on the right. A mouse actually has slightly more genes than we do. A tiny little mouse, more genes. A mosquito and a fruit fly have slightly fewer, not all that many fewer genes than we do. Here's our pufferfish, right about kind of the same level as us. So a pufferfish has the same number of genes as we do. And you can see some plants have way more genes than we do. This is why plants are kind of tough to work with. Like I said in the last lecture, they just are really complicated. So the number of genes, the genome size doesn't really relate directly to how complex an organism is, because I think we'd all agree, at least from a human standpoint, that we are more complex than a pufferfish or a mouse. I guess there's a philosophical argument probably to be made there, but from a biological complexity standpoint, we are more complex, but it doesn't mean we have to have more genes to make us more complex, which is pretty interesting. And like I said, that was just a reference for one human, basically. Now the goal is to look at the genetic diversity of the human population. Obviously, we don't have the same genomes. Everyone has a different genome. So the 1,000 Genomes Project came along, where they're looking at, the goal is at least 1,000 individuals to produce, I guess, comprehensive, but not totally comprehensive, but fairly comprehensive catalog of all of the variation in human genomes. So they looked at 26 different populations. Again, there's far more than 26 populations on the Earth. So this is just a start. But with that, they identified 80 million genetic variants. So with these 1,000 individuals, there's 80 million different variations within our genomes across those 1,000 people. So diversity is kind of the next goal for these genome projects, figuring out how we differ and how that might affect our health. Some more kind of technical stuff. So if you know the base pair sequence, the A's, the G's, T's, etc., it doesn't really tell you about what the genes do. So another step forward would be to actually annotate. So genome annotation is going to assign functions to these sequences. So they can figure out what the function of a certain sequence is. What protein does it code for? What does that protein do, etc. That's a lot more useful than just knowing a bunch of letters in a row. So there's a few databases. The National Center for Biotechnology Information is kind of the big one, NCBI, where they collect all of these sequences and then slowly over time people annotate them. So they determine what the functions of these different sequences are. And then say you're looking at a sequence of some organism, maybe a human, maybe a pufferfish, you can try to match your sequence to one of the sequences in these databases. And then if it's annotated and they already know what the function of that sequence is, then you can figure out for your organism what the function of that sequence is. So it's a great reference. There's great referenceability. So there's been a big push to make a lot of this public and freely available. So to any researcher, kind of as a collaborative effort. Genomics is typically divided into sort of two different subfields. So you have the comparative side and the functional side. They are exactly what they sound like. So comparative genomics compares genomes between species. So we have about half of the genes between humans and fruit flies are 50% similar, which is kind of crazy. The tiny little fruit fly, if you've ever seen those in your house, they have half of their genes are the same as ours. So we know a lot about fruit flies. They're an important kind of lab experimental model. We know what a lot of their genes do, so we can compare their genome and their functions of those genes to our sequences and potentially understand a little bit more about what our genes are doing as well. So that's comparative genomics, using what you know about one species and applying that to another species based on just the genomic sequence. This is called, this uses something called synteny. So synteny is where there are similar DNA arrangements between different species. So like I said, half of our genes would be considered syntonous with fruit fly genes. So we can compare those that are the same. Obviously the ones that aren't the same, we'd have to use a different organism for a comparative standpoint. But that's the idea behind comparative genomics. Take something you know about and compare something you don't know about, and see if there's similarities there. And then the functional side is exactly what it sounds like, figuring out the functions of the genome, so specific genes within the genome. A big part of this is proteomics, which is figuring out what proteins are coded for by specific genes. Proteins are tough. They're a lot harder to study than just DNA because they're a lot more complicated. DNA is as complicated as it may seem to us, just two strands of a bunch of nucleotides, right? Proteins, so we ended translation talking about an amino acid strand. So we just had this long string of amino acids at the end of translation. After that, a lot of stuff happens in order to make it a functional protein. It folds over on itself, it has bonding within itself, it might bond to another protein. They can get really complicated. So those post-translational modifications, anything that happens after translation that makes the protein have the function that it has, is really difficult to understand. It's not just the sequence of amino acids, it's all of that other folding and bonding within it that actually makes it functional. There's also what we call isoforms of proteins, where those non-coding sequences, so we talked about the mRNA at the end of transcription, so backing up to transcription, a bunch of stuff is taken out of the pre-mRNA to get that final mature mRNA. That happens differently depending on the protein, and it might not change the function of the protein, but it's going to change the base pair sequence, which makes it a little bit more difficult to study proteins from that perspective as well. So functional genomics, take home, it's looking at the function of the genes. Comparative genomics, you're comparing between different species to figure out, to maybe look at the function of one gene where you know that in one species, but you don't in the other. Those are the two main divisions of genomics. Another important term, a field, I guess, that includes, it's kind of above and beyond genomics. It's above biology, it's above any individual field. This is a really interdisciplinary field that's huge and growing these days. So if you're interested in biology and computer programming and modeling, this is the field for you. Bioinformatics. It basically takes a ton of data and analyzes it using computer programs, really complex models, and applies the biological aspects to it. So yeah, it's a huge field. If you're good at incorporating a lot of different topics or you like interdisciplinary stuff, this is a really interesting field to be in. So yeah, you can, I mean, among a million other things, it can be used to compare a protein sequence from something you already know, like an already known amino acid sequence that creates a protein, and compare that with something that's maybe unknown and see if those functions match up. So yeah, bioinformatics, you'll probably hear about that going forward, especially if you stay in any kind of science, you'll hear about bioinformatics. All right, so that's the basics. Let's talk about applications. I think applications are some of the more interesting aspects of this biotechnology and genomics section of the course. This one, maybe not so much, but this is where they started. They tried to create a synthetic cell, which what is a synthetic cell? They basically sequence the genome of the smallest bacteria, this little guy, I guess. I mean, that's what popped up. That's what's shown in the book. So this tiny bacterium, they sequenced the genome and then chemically recreated it. So they put all of those nucleotides together to create the genome. Then they inserted that genome into another species of bacteria to see if it would actually control the growth of that second species of bacteria, and it did. So they genetically engineered a synthetic genome, put it in an actual species, and it controlled the growth of that species. Not super applicable, but interesting kind of in and of itself. But that was sort of the starting point for applying some genomic technology. So it's a synthetic cell. All right, so let's talk about medical applications. Medical applications, I won't really get into forensics. I'm going to show you guys this video, which is a really interesting video about medical applications of genomics. We've talked about some of the biotechnology stuff with medicine that's been done. I have 2 bullet points here, which seems kind of ridiculous for the medical applications of genomics, but these are 2 of the probably the most important applications where this genomic technology can be applied. Looking at genetic abnormalities, which this guy will talk about in this video, along with determining the genetic basis of human diseases. So again, in order to use that CRISPR system that I was talking about, you have to know where the problem is first. So you have to find the part of DNA that's kind of messed up and not coding for the right protein, and then you can go in and fix it. But genomics has to come first to figure out where those issues might be. Forensics, I mentioned with DNA fingerprinting. Now they can do much more high-tech kind of identification of victims or of suspects by looking at their genome rather than just a fingerprint. Okay, so let's watch this video. This is from 2011, so it's already 10 years old, which is kind of crazy. And the guy who's talking is an owner of a biotech company. He's a really smart guy who just explains genomics and biotechnology kind of all at once in a really interesting way with some great examples. So let's watch this. Remember, this is from 2011. So the amount of progress that's been made since he gave this talk has been huge. But even this sounds kind of futuristic. It's pretty amazing. Okay, I don't think we need that. Come on.
Ladies and gentlemen, I present to you the human genome. Chromosome 1, top left, bottom right are the sex chromosomes. Women have 2 copies of that big X chromosome, men have the X and of course that small copy of the Y. Sorry boys, but it's just a tiny little thing that makes you different. So if you zoom in on this genome, then what you see, of course, is this double helix structure, the coda-like spelled out with these 4 biochemical letters, or we call them bases, right? A, C, G, and T. How many are there in the human genome? 3 billion. Is that a big number? Well, everybody can throw around big numbers, but in fact, if I were to place one base on each pixel of this 1280 by 800 resolution screen, we would need 3,000 screens to take a look at the genome. So it's really quite big. And perhaps because of its size, a group of people, all by the way with Y chromosomes, decided that they would want to sequence it. And so 15 years actually, and about $4 billion later, the genome was sequenced and published. In 2003, the final version was published, and they keep working on it. That was all done on a machine that looks like this. It costs about $1 for each base, very slow way of doing it. Well, folks, I'm here to tell you that the world has completely changed, and none of you know about it. So now what we do is we take a genome, we make maybe 50 copies of it, we cut all those copies up into little 50 base reads, and then we sequence them massively parallel. And then we bring that into software, and we reassemble it, and we tell you what the story is. And so just to give you a picture of what this looks like, the Human Genome Project, 3 gigabases, right? One run on one of these modern machines, 200 gigabases in a week. And that 200 is going to change to 600 this summer. And there's no sign of this pace slowing. So the price of a base, to sequence a base, has fallen 100 million times. That's the equivalent of you filling up your car with gas in 1998, waiting until 2011, and now you can drive to Jupiter and back twice. World population, PC placements, the archive of all the medical literature, Moore's Law, the old way of sequencing, and here's all the new stuff. Guys, this is a log scale. You don't typically see lines that go up like that, right? So the worldwide capacity to sequence human genomes is something like 50 to 100,000 human genomes this year. And we know this based on the machines that are being placed. This is expected to double, triple, or maybe quadruple year over year for the foreseeable future. In fact, there's one lab in particular that represents 20% of all of that capacity. It's called the Beijing Genomics Institute. The Chinese are absolutely winning this race to the new moon, by the way. What does this mean for medicine? So a woman, stage 37, she presents with stage 2 estrogen receptor positive breast cancer. She is treated with surgery, chemotherapy, and radiation. She goes home. 2 years later, she comes back with stage 3C ovarian cancer, unfortunately treated again with surgery and chemotherapy. Comes back 3 years later, age 42, with more ovarian cancer, more chemotherapy. 6 months later, she comes back with acute myeloid leukemia. She goes into respiratory failure and dies 8 days later. So first, the way in which this woman was treated in as little as 10 years will look like bloodletting.
Do you guys know what bloodletting is? Okay, I was like, I don't know if people know what this is. So back like in medieval times, they used to cut you open and let your blood flow out because they thought that would get rid of the disease that you had. So obviously we know that's not the way to get rid of a disease now. So this guy's saying that chemotherapy, surgery, radiation, those are basically going to look like how we view bloodletting from the Middle Ages. Anyway.
And it's because of people like my colleague Rick Wilson at the Genome Institute at Washington University who decided to take a look at this woman post-mortem, and he took skin cells, healthy skin, and cancerous bone marrow, and he sequenced the whole genomes of both of them, right? In a couple weeks, no big deal, right? And then he compared those 2 genomes in software, and what he found, among other things, was a deletion, a 2,000 base deletion across 3 billion bases, right? In a particular gene called TP53, if you have this deleterious mutation in this gene, you're 90% likely to get cancer in your life. So unfortunately, this doesn't help this woman, but it does have severe, profound, if you will, implications to her family, right? I mean, if they have the same mutation and they get this genetic test and they understand it, then they can go and get regular screens and they can catch cancer early and potentially live a significantly longer life. Let me introduce you now to the Beery twins, diagnosed with cerebral palsy at the age of 2. Their mom is a very brave woman who didn't believe it. The symptoms weren't matching up. And through some heroic efforts and a lot of internet searching, she was able to convince the medical community that in fact they had something else. What they had was DOPA responsive dystonia. And so they were given L-DOPA and their symptoms did improve, but they weren't totally asymptomatic. Significant problems remained. Turns out the gentleman in this picture is a guy named Joe Beery, who was lucky enough to be the CIO of a company called Life Technologies. They're one of the 2 companies that makes these massive whole genome sequencing tools. And so what he did was he got his kids sequenced, and what they found was a series of mutations in a gene called SPR, which is responsible for producing serotonin, among other things. So on top of L-DOPA, they gave these kids a serotonin precursor drug, and they're effectively normal now. Guys, this would never have happened without whole genome sequencing. And at the time, this was a few years ago, it cost 100,000. Today it's 10,000. Next year it's 1,000. The year after it's 100, give or take a year. That's how fast this is moving. So here's little Nick. Likes Batman and squirt guns, and turns out Nick shows up at the children's hospital with this distended belly, like a famine victim. And it's not that he's not eating, it's that when he eats, his intestine basically opens up and feces spill out into his gut. So 100 surgeries later, right, he looks at his mom and says, "Mom, please pray for me, I'm in so much pain." His pediatrician happens to have a background in clinical genetics, and he has no idea what's going on, but he says, let's get this kid's genome sequenced. And what they find is a single point mutation in a gene responsible for controlling programmed cell death. So the theory is that he's having some immunological reaction to what's going on to the food, essentially, right? And that's a natural reaction which causes some programmed cell death, but the gene that regulates that down is broken. And so this informs, among other things, a course of treatment for bone marrow transplant, which he undertakes, and after 9 months of grueling recovery, he's now eating steak with A1 sauce. The prospect of using the genome as a universal diagnostic is upon us today. Today, it's here. And what it means for all of us is that everybody in this room could live an extra 5, 10, 20 years just because of this one thing, which is a fantastic story, unless you think about humanity's footprint on the planet and our ability to keep up food production. So it turns out that the very same technology is also being used to grow new lines of corn, wheat, soybean, and other crops that are highly tolerant of drought, of flood, of pests and pesticides. Now look, as long as we continue to increase the population, we're going to have to continue to grow and eat genetically modified foods. And that's the only position that I'll take today. Unless there's anybody in the audience that would like to volunteer to stop eating. None, not one. This is a typewriter, a staple of every desktop for decades, right? And in fact, the typewriter was essentially deleted by this thing. And then more general versions of word processors came about. But ultimately, it was a disruption on top of a disruption. It was Bob Metcalfe inventing the Ethernet and the connection of all these computers that fundamentally changed everything, right? And suddenly we had Netscape and we had Yahoo and we had indeed the entire dot-com bubble. Not to worry though, that was quickly rescued by the iPod, Facebook, and indeed Angry Birds. Look, this is where we are today. This is the genomic revolution today. This is where we are, okay? So what I'd like you to consider is what does it mean when these dots don't represent the individual bases of your genome, but the connected genomes all across the planet? So I just recently had to buy life insurance, and I was required to answer A, I have never had a genetic test. B, I've had one, here you go. And C, I've had one, and I'm not telling. Thankfully, I was able to answer A, and I say that honestly in case my life insurance agent is listening. But what would have happened if I had said C? Consumer applications for genomics, they will flourish. You want to see whether you're genetically compatible with your girlfriend? Sure. DNA sequencing on your iPhone, there's an app for that. Personalized genomic massage, anyone? There's already a lab today that tests for allele 334 of the ABPR1 gene, the so-called cheating gene. So anybody who's here today with your significant other, just turn over to them and swab their mouth, send it to the lab, and you'll know for sure. Do you really want to elect a president whose genome suggests cardiomyopathy? Now think of it, it's 2016, and the leading candidate releases not only her 4 years of back tax returns, but also her personal genome. And it looks really good. And then she challenges all of her competitors to do the same. You think that's not going to happen? You think it would have helped John McCain? How many people in the audience have the last name Resnick like me? Raise your hand. Anybody? Nobody. Typically, there's one or two. So my father's father was one of 10 Resnick brothers. They all hated each other, right? And they all moved to different parts of the planet. And so it's likely that I'm related to every Resnick that I ever meet that I don't know. So imagine if my genome were de-identified, sitting in software, right? And a third cousin's genome was also sitting there, and there was software that could compare these two and make these associations. Now I have to imagine my company has software that does this right now. And so imagine one more thing that software is able to ask both parties for mutual consent. Would you be willing to meet your third cousin? And if we both say yes, voila, welcome to chromosomally linked in, right? Now, this is probably a good thing, right? In bigger clan gatherings and so on, but maybe it's a bad thing as well. How many fathers in the room? Raise your hands. Okay, so it just turned, experts think that 1 to 3% of you are not actually the father of your child. Look. These genomes, these 23 chromosomes, they don't in any way represent the quality of our relationships or the nature of our society, at least not yet. And like any new technology, it's really in humanity's hands to wield it for the betterment of mankind, or not. And so I urge you all to wake up and to tune in and to influence the genomic revolution that's happening all around you. Thank you.
So Casper finds a thing and this like locates where the actual, so do they use it together?
Yeah, like I said, so in order to use CRISPR, you need to know where the issue is. So he was talking about people doing like sequencing an entire genome. They found where there was like a massive deletion. So that's where they then aim CRISPR at to go kind of rebuild that. Yeah, it's amazing. So yeah, that was 2011. It is now 2021. So I think it was, I don't even remember how many years ago it was, but I remember hearing Angelina Jolie got her genome sequenced and found out she had the cancer causing gene. So I think she had a double mastectomy in order to avoid getting breast cancer. So it's already being used to kind of avoid certain diseases and hopefully to fix certain ones. That little, God, I cry every time I hear about that little kid and with the distended belly. And now he's basically eating normally. It's awesome. Okay, so just a few more, well, basically, I just want to talk briefly about some ethical issues because I think those are fascinating. So there's obviously ethical issues that are going to come along with some of this genomic stuff. Like he was talking about his life insurance agent wanting to know about, if he'd had a genetic test, if it said anything bad, whatever. So I just have a couple interesting stories here about patenting human genes. So there was a move to potentially patent human genes by Myriad Genetics, and they tried to patent essentially 2 genes that were known to cause breast cancer. That would give them exclusive rights to test and then to develop drugs for breast cancer. Obviously, that has huge economic ramifications and implications for them. The Supreme Court, I think, thankfully, denied their patent request, and they said you can't patent natural gene sequences. So my gene sequences, I guess I essentially created, so they can't patent, some other company can't patent that.
What do you mean by patent?
Basically have rights to it, so no one else could work on those gene sequences. Like if you invent something, you can get it patented. Like a lot of the vaccines and stuff are patented for a certain period of time, so no one else can develop them. So the drug manufacturers make money off of those.
So it's like a monopoly.
Yeah, exactly. I just heard that on the news this morning. It's basically a monopoly. So yeah, natural gene sequences can be patented, but human altered ones can. So those Roundup ready crops that I talked about that are drought resistant or that are herbicide resistant, those can be patented. Monsanto patented that gene because they made it. So that's the main difference right now between patenting and not patenting genes. So probably, I guess for now, the right direction, hopefully. And, well, I don't really want to talk about this, but just really quickly, you might have heard of people kind of, or scientists recreating deadly viruses. So they do this in order to be able to study them. The Spanish flu is one of them that happened in 1918. Smallpox, I think, is another one. These have been eradicated, so they're not circulating in the world, but they're in these super high security labs in order to study them. So that's an ethical issue, like, should we recreate them? Should we not recreate them?
How do they recreate it? Do they have like a little bit of the virus to recreate it?
Yeah, so they sequence the genome. I'm not sure where they get the virus from. That's a good question. I think smallpox, they still had some, but it got eradicated in the human population. But I'm not sure about the Spanish flu because that was before they probably had those abilities. And then finally, have you guys heard of 23andMe, the company? So I always see these ads around like Christmas. I guess people want to get their genome sequenced as a present. So you can get your whole genome sequenced with there's other companies too, but 23andMe is kind of the most popular one. And basically what they do, you swab your cheek, send it off, and they'll sequence your genome for you. And then they'll send you information about your genome. So if you have any like maybe unknown health issues, so if you're prone to high blood pressure, or if you're prone to obesity or something like that, they'll tell you about that. And then the important part is, and the marketing part is, they sell you products based on that. So this little, you probably can't see this little pink bubble here, but it says health and ancestry. So it'll tell you about your ancestry, which is interesting. And they'll also tell you about your health, like I said. So anything maybe that isn't ideal, and I'm sure we all have stuff in our genome that's, you know, not ideal and not ideally healthy. So they try to market and sell you health products based on that. Currently, they can't share that info with any third parties. So they have to keep it secure and they can't sell it. But I mean, privacy issues, even just with social network sites, have become a huge issue. So I think this is probably going to be front and center as more and more people do it, and they kind of move further in the direction of maybe selling some of your information if you do this. A heads up, if you search for 23andMe, because I just searched for an image of it, they will bombard every website that you go to from then on. I still am getting ads for 23andMe just for looking for a picture for this. So anyway, they're good at retargeting essentially in their marketing scheme. So anyway, those are a few ethical issues of genomics to think about. I'm sure there's a million more that I don't really have time to cover. But yeah, I think that kind of wraps up the genomics lecture and the material. So we're not doing, I have genome evolution on the schedule. We're not doing that chapter. So was it just biotechnology and genomics? I think those are the only.
On the study guide?
Yeah, what was the other one? There's another chapter, right? What am I forgetting?
Yeah.
Anyway, we're not doing genome evolution on the next exam or on the final. Okay, and I'll see you guys on Monday for the review. That is it.
She was like,